Okay, so today is week 3, but we, because of this winter storm, and we missed one class lecture last week. So this will be lecture 2. I'm going to talk about the vectors and matrices. So this is the outline for today's topic. The most important, the 3 items: the multiplication of matrices, and the geometric interpretation of matrix vector multiplication, and then I will introduce the eigenvalue, eigenvector concept. We plan to have the cell ranger discussion on how to prepare this, but last week the computer session, Shura has already demonstrated the pipelines to you, and I just will be very quickly going through if we have time at the end of the lecture, okay? I want to let you know first, you have to be aware there another world of the research. We are doing biomedical research. We are working in the living organisms and working on the animal, humans, microbiome. But the other domain of knowledge is from the computation, engineering, people in computer science. They deal with the data. If you open the paper and you read what they write on the papers, it's very, very different with biology paper you read. When they talk about the distribution, when they talk about the normalization, when they talk about the model or modeling, you have a different mind with, they have a different mind with us. So I hope through this interface lecture, you cross the boundary which between biology and computer science, data science, a little bit cross to their field and feel what they are thinking and what's the language, what's the symbolic system they are using and why they use it in that way. For example, recently I read a paper, I discussed gene and interact with others. I will write some like a gene A interact with the gene B. So it's just a very natural language, like print English. But for other people, if they from a computer science background, they probably will see or use some symbol like a mathematical label, say gene XI interacts with gene XJ. They have a different ways of thinking and different ways of describe what they want to see. In some sense, I want you to become more appreciate what the system they are using. So that's the interface. We talk about the vectors and the matrices. That's what we can have this terminology in us to get into the field of their world. By the way, this is a huge gap and from biology to computer science, bioinformatics. The gap is much bigger than you thought, okay? So first, let's see. Have you ever heard about, for example, DeepSeek? I don't know if you follow the news, tech news. DeepSeek, nobody? DeepSeek, okay. DeepSeek, okay. 3, so 3 against 2. OpenAI, who heard about this? Okay, everybody. ChatGPT, okay. Another I prepared is this. This has been, I remember this very recently, but if I check, this already 2022, about 2 years. There's a breakthrough that year. I remember very clearly because I announced this in the lecture as well. So AI found a new way to do matrix multiplication. Okay? If you know how to do matrix multiplication, you have to multiply every element between 2 matrices and sum it up at the end. It had been, you can calculate by hand, okay? But the AI found a new way to do this multiplication and to save several steps in the middle of this operation. why it's a big deal? Why it's in the headline about all of these science articles? So I can quickly show you some of those algorithms they use. Okay, so a new algorithm developed by the AI to search and to save one of the steps in the multiplication of matrices in the middle of calculation. It's a big deal because the whole AI machine learning field, everything is rely on multiplication of matrices. If you can save just one tiny step in the middle, you save huge of computational power you need to achieve the same results. Okay, so everything you can think about, the neural network, ChatGPT, OpenAI, DeepSeek, whatever. At the very end, you are doing matrix multiplication. All the neural networks, how they evolve, how they develop, how they learn things, it's by this. Okay, so that's why it's a big deal. All right. So vectors and the matrices, when we deal with this, we can see, okay, we play with Excel spreadsheet. This is a matrix, okay? But those are really, really important. This is a part of this subject in linear algebra, as I said in the first lecture. Linear algebra is all the foundation of you need to do machine learning. So every step we are working in the single cell data analysis using machine learning, we rely on the operation of linear algebra. All right? For instance, we want to show, this is a PCA basically. I don't know if you can play this. So this operation, if you have been given a bunch of data in the 2D, or you want to find a straight line and try to go through this cloud of data points, blue ones, you use linear algebra operation. Basically, you use multiplication of matrix to rotate this angle of the line to go through this cloud of data. And you can achieve certain object. So if your objection is to, your goal is to minimize the distance from all the data points to the line, sum together. So you have overall distance from every single data point to the line, overall minimized. Okay, you obtained an optimized solution of this straight line which goes through all the data points. Okay, so this is like one of the operations we will certainly do in using matrix multiplication. It's as one of the step. What's an application for this? If you, I gave you an object, in this case as a teapot. I got this from a world market. I don't know if this link still works. But to give you the hypothetical situation, I only allow you to show one picture and which angle you want to use to show people the most feature of this teapot. You probably will see like this angle. The reason is you show people the most dispersed feature points on this object. You don't want to show it from the bottom, or you want to use this angle because that will allow data point to superimpose onto each other. You lost the information. You want to display this. So the same idea. Here you want to find a way to display all the data points from certain angle to showcase from lower dimension, because this 3D become 2D, you want to use 2D to represent the most feature information in this object. So you will have to rotate your angle. Okay? Make sense? All right, so lineage algebra and this matrix multiplication will tell you how to rotate it, and their object function to tell you when to stop. Okay, so we are going to rotate this, but this is only 3D. The data you are going to play with are the single-cell RNA-seq data. We consider it to be higher dimensional data. It's more than 3, 4, because each cell you define it with thousands of genes. So if those genes are independent, considered to be independent to each other, then we are using thousands of dimensions of information to define each cell. So we are not dealing with the T part of 3D, we are dealing with object in the higher dimension, thousands of dimensions. So, but still the same principle apply here. We are going to rotate this complex cell population manifold, okay? And to find a certain angle to look at this, okay? So that's what the PCA will do. So there are many, many resources online. So you can have textbook. There are many, many excellent textbook, and they are free as well. There are so many YouTube channels will discuss this. What I can present here is a more condensed, more customized learning experience. What I really want is to have my configuration of my understanding and directly put in your head, if that's possible. But because of the limitation of this information communication, I can only using the language to describe what I'm going to see and have to transfer the understanding on my head, in my hand to your mind. Okay? I hope in the future the AI will have some other solutions from Neuralink, for example. I can just copy it, then you are done. That will be the best situation. So that's why this AGI, so it's artificial general intelligence, will be successful, because every single skills or mindset learned by one machine will be able to easily copy and propagate into the cloud or other machine. So that increase is exponential, and not as a human communication. We know so little about this unknown world, and there. So you still don't know what's eigenvalue, for example, until now. I assume how many people know here? Eigenvalue, eigenvector, what's that for? I hope at the end of these 2 hours you know a little bit. But that's the transformation of the construction of mind, of information from here to there. That's what I try to achieve. I will try whatever approach, okay, we can to achieve that. So don't worry about you get the degree, the grade A or B, or nobody really care at the end. So to have your mind change is the most important. Okay. Very simple. Scalar is a single number. Okay. If you have a equals 5, then 5 is a scalar value. So that's, you can consider a scalar number as a 1, 1, 1 matrix. That's a dimension is 1, 1. Then the matrix, for example, A, this matrix has 2 rows and 3 columns. This is a 2 by 3 matrix. A vector is one of the dimension of matrix is 1. So if you have m by 1 matrix, this is a vertical matrix vector. So small a, so you have a column of numbers. Okay. If you have a row matrix, that will be 1 by n matrix. So the first dimension is 1. So this is just a row of data. Okay. And this particular symbol here is a T, okay, capital T in the let's see. We use this T to transpose, okay? So if A is a row vector, so you have x1, x2, xn, then you transpose this to become column vector 1, x2, okay? So in computer science, in data science, people use column vector as a data, okay? So this will be, by default, we will use this to encode information. So this is a variable, so this variable, you can consider this as an object, a data point, or maybe a cell, and the value are the parameters values to define this particular object here, okay? But in statistics, people use row matrix to see, okay, this is one of the record. They use row matrix, row vector, but in computer science, in data science, people use vector, column vector. Sometimes people get confused. If I only gave you a small a, you don't know whether it's a row vector or column vector, okay? You have to find a way to do it. I believe in quantum world, people use this as a row vector and then use this as a column vector. So they invent their own symbols or notations to differentiate the difference of these two. Okay, so if you have a 2D vector here, then you can represent this in this space. Okay, so you can visualize this. I can change the value here using different notation x, y. So here you have a vector, and its coordinates is x and y. So that's where x is, and there's y. Okay, so this gave you the geometric interpretation of vector. We can represent 3D up to 3D vector using this graph. If they're 4D or higher dimension, you have to imagine, okay, in the higher dimensional space. There's n-dimensional space, nd space. And this vector is in that place. Where it is defined by those values. You can think about this as steps. You move through the first dimension as a step x1, and then step x2, step in the third dimension x3. So you move until you reach the position where all this value define where this particular vector is located in those high dimensional space. Is that clear? Okay. There's no much good way to visualize the high dimensional. So in this plot here, I believe it has 3D, 2D example, 3D, and try to use this to represent 15 dimensional space those values, okay? So we try to reduce the dimension to a higher dimensional object and reduce to 2D or 3D so that you can visualize your cell, for example. Okay, so if I have this vector, A, or maybe another, B, so what we can do? We can do scalar multiplication, addition, and measure the length of unit vector, okay, to compute how long this is, to multiplicate A by 3, for example. After that, you will have a 3 times longer vector if you do this scalar multiplication, right? We will later discuss about this inner product, okay? So I will leave this later. Once we know how to do the matrix multiplication, then we see how can we multiplicate A by itself transposed, okay? So, but this is follow the general rule of the matrix multiplication. So if you know how to do matrix multiplication, you will know how to multiplicate these two, because small a is just a special case for the big A, okay? If you know how to do matrix multiplication, and you will know how to do this, and this is so-called inner product or dot product. So don't worry about it for now, okay? So sometimes people will be giving a vector which not start from the origin, okay? So if someone gave you a vector, so from a P to Q, you can easily move back to here. So this should be the same in theory, because you just change nothing but only your perspective of where you think about as an origin. So this will be the same as this. You just move where your origin is. So you can do this transformation to move that back to the origin. Not too hard, right, so far? Okay. So this is demonstrate the geometric interpretation of addition and subtraction of two vectors. If you have vector A and B, A is in the position 3, 4, and B is in the position 7, 2. So you put, so this is writing in a column, okay, in the vertical vector form. So you have one vector, another, you add them up. You can do the first dimension, the two values add together, and this. The interpretation of this is you calculate the vector which pointed to the middle of this, okay? When you do subtraction, so this is example of using this minus that, and you obtained this one here, all right? And this one can be represented, depends on you use this minus that or that minus this. The end result will be either as a u minus v or v minus u. Okay? This is useful if you want to calculate, for example, the distance between these two data points. So how far away from A to B, what you can do is to use the vector A, which is the data point A minus data point B. And then you obtain this vector, and then if you know how to calculate the length of the vector, then you get the distance between 2 points. All right, so this is a very basic operation to do the subtraction of two vectors and then get the distance between 2 data points represented by these 2 vectors. Questions? Okay, yeah, so far so good. So your brain is still clear, right? Good. Okay, so come to here, the dot product. So depends on how you write this. The dot product, for example, in this case, the 3D example. So if I use the column vector, then you should transpose this first. Transpose this, then, yeah, so this is the correct way to write, because if you flip this around, you got a different, it's not inner product, it's called outer product, okay? So just make sure you know this will be a column vector multiplied by the row vector by transpose it, the same, then you obtain a single value here. So I just show you an example here. We will spend more time next week to talk about the interpretation and use of this equation here, okay? So so-called dot product, it means if you have the operations and you put a dot, a thick dot in the middle to indicate I'm doing the A times B. And this dot, if people know you are doing the dot product, they will know you assume you are doing this so-called inner product, even though you don't know how you put this A and B, the dimension. So because if you only write small a, small b, you don't know whether A is horizontal or vertical vector. So in this case, if you know it's dot product, you don't care. As long as you put this into a correct dimension, you will get a single value at the end. So that's why people use dot product, just ignore to know whether A and B are the vector or horizontal or vector. So at the end, you get a single value. So you basically smash down the product into a single value. So the geometric meaning of this single value is the length of vector x, so how long this part is, times how long this part is, because lengths are just scalar, just value, right? So the multiplication of these two will still be a single value. And then times the cosine of the angle between these two vectors. All right? So all you need to know right now is the dot product or inner product of two vectors is a scalar value. There are some articles online to explain what this is, how this will be achieved by using some transformation of very basic geometric operations. and then to get this equation here, okay? Okay, move forward. We need to know what is norm. What is the norm for a vector? So this A vector, so you have x or y. So the norm is, there's a symbol people use double, how to say this? Then you put your vector in the middle, and then to double this vertical line around to show, okay, this is a norm. The norm is how long this is, okay? So to calculate this, you have to use squared plus y squared, then that's norm A. Is that clear? Is a Pythagoras theorem, right? So you have the coordinates, basically how long the x is, how long the y is, then this is just how long this distance. This is also called L2 norm, so, or sometimes 2 norm, because there are other definitions of norm. For example, L1 norm will just be x plus y, maybe. So you have to check the definition, but this is a definition of regular. If you don't write anything around this double vertical bar symbol, then we're talking about L2 norm, and that's basically how long this vector is. In the example here, you have 3D example. If you have a vector in the 3D, you apply this Pythagoras theorem again, and it's applicable. The beauty of this theorem is you can apply to even higher dimension. Just use the same form of this equation. You get the norm of vector, no matter how high the dimension is, okay? So to be specific, sometimes people write, put a small 2 here. So the reader will know, okay, you are using L2 norm, which is how long this vector is. Or you may be more specific, you put 1 here, people know, okay, this is 1 norm, you just multiplicate, plug all this together. I don't remember whether you need an absolute value or not. Maybe you should, yeah. So we can look at L1 norm definition. Maybe you have just add them up, basically. If you leave this nothing, then people will assume this L2 norm, okay? So this is just some notation people will definitely use in computer science, in machine learning field. If you read paper, I would see maybe half of those papers will have something like this if they discuss the objection of the normalization, the optimization. excuse me. So we don't have to discuss this for business norms because this is for matrix. Okay. Unit circle. So you have this coordinate system, x, y axis, then you define a vector here. So for example, this is simplest vector, have a position at 1, 0, okay? And this one, zero, 1. So for this two vector, you know their norm is 1. So you try to find all the vectors in this space with a norm equal to 1. And all these vectors form a circle because from 0 to any position in this circle, you get a vector have a norm equal to 1. So this circle is called a unit circle. It tells you the direction of your directionality, basically, of where the direction is of your vector on this circle, but there's no length. They're unique lengths. Okay. So for instance, and where what this point should be, if it is 45 degree, I would see so x squared plus y squared, and this should be equal to 1. So this position, what that should be? So x, y should be y should be square root y, right? So if I want to know the coordinate of this position, which is 45 degree in that upper right, this vector, unit vector, should be 1 over square root 2. In order to satisfy this equation, the norm is 1. Okay? You can compute whichever unit vector as long as you know the angle you are using is theta here. The equation is there. Okay? Why is this circle important? It's define the direction of your vector where they point to. If the direction is 0, you point to the right, straight right. If you point to straight up, you know it's 90 degree up. If the 3D, you define a sphere, you need 3 sphere. If you hide D, you still have your imagination sphere like this, you know the unit vector point to that particular direction you in the high dimension. All right? So we will utilize those vectors, unitary vectors, later. Okay? Unitary means their lengths are the unit 1. That's all the norm of those vectors are 1. So why is it useful? It will tell you the direction. If you do operation using this to your data point, it will not change the scale, not change the magnitude of your data point, because your data point is also a vector. So if I multiplicate my vector from unit circle to your vector, I only change your direction, not change your magnitude. Will not change your original data points value, only change. So that's a rotate of your t-port later, okay? I'm going to rotate your t-port for some of this using unit vectors, change your angle of the road, but not change the shape of your t-port. Okay, so matrix operations. We can have transpose, okay? So if you have n by m matrix and you transpose, become m by n. Identity matrix means everything are 0, only the diagonal position are 1. You can add, you can do the scalar multiplication. So all the matrix elements multiplied by a single number. Then we will discuss how to do this correctly, and then if you know how to do matrix multiplied by another matrix, and then you will know this as well, because vector is just special matrix. And this is a little different, this is different, this is very different, okay? That's something different. Transpose is easy, and it's easy, maybe not. Yeah, so you got to remember, once you transpose, you put columns become rows, rows become columns. You can do this in the Excel spreadsheet. You know, you select a table, then you copy. You want to paste, you can selection, transpose that table, and the table column becomes rows. And in MATLAB, we just put a matrix, and then the function trying to transpose all. There's an easy way to just put a apostrophe on the top of this matrix. This operation will transpose your matrix. This is so commonly used. So all the languages should have that function easily built, and some put this super script T to indicate the transpose, which is here, okay? This is just transpose that vector, but the same as to transpose the matrix as well. This is identity matrix, looks like, and this is only 4 by 4. So identity matrix is supposed to be squared. So, well, not necessary. Sometimes you have also, you can truncate this and make it become, for example, 4 by 3, and this part is still identity matrix. It's just not complete, okay? Adding operations for the matrix, follow this general rules. You can switch the order of A plus B or B plus A the same, because you just add up all the values inside the matrix. In this case, A and B has to be the same dimension, so A is a 3 by 2, then B has to be 3 by 2, otherwise you don't know how to add them together, all right? Nothing special, okay? Everything clear so far. Scalar multiplication. In this case, this example, and you multiply a scalar, a number 2, and every element inside the matrix got the double their value. Okay, so if you know this, you can demonstrate all these cases are true, because their addition operation for A and B, and before and after you do this scaling, it will be the same. Okay, if you do 0, then it becomes 0. Okay, so don't have to remember anything. It's just common sense so far. Okay, so matrix, one matrix multiplied by another matrix. Just remember, in general, this is not commutative. You cannot say A multiplied by B equal to B multiplied by A. This is not the case in general. So this example is to show you how to do this matrix multiplication. I want to know how many of you have ever tried or know how to do this. Just have ever, you learned that before, you know that basically. With some of you, yeah. Okay, so this I put a start here. So this I believe this is an important slide to. Just one slide will teach you how to do this and what also imply what that means here. Okay, so we will spend some time on this page. So if you forgot a lot of the content of this lecture, but remember this. Okay. Because this is a good example to demonstrate how you can do it and what's the meaning of the matrix multiplication as well. I will also let you later to think about how to do the opposite. If you have the end product of this A and B multiplication, whether you will be able to reverse to back to A and B. If I'm not telling you what A and B look like, there are some applications in single cell studies as well. Not some, there's many good applications for this procedure, all right? So if you have time, just read the text, and it's a small story, and you and your roommate try to make breakfast sandwich, and there are three different type of sandwich. And you and your roommate each want to make different numbers of three types of sandwich. So that's why there's matrix A to tell you, oh, you want to make 4 sandwich type 1. That's the number here. You want to make 5 sandwich type 2, 3 type 3, and your roommate want to make 3 different sandwiches, but different numbers. Okay, so that's what you want to do. Okay, so the first, this A matrix. Have the number 4, 5, 3. Second, have a 3, this is you, and then that's your roommate. Okay? So I'm going to put this in the special location. So when you do this matrix multiplication, try to remember to put in the same way I'm going to put, so you can always get the correct results at the end. So I put my first matrix here, my second matrix in this corner here, and remember this would be 3 columns, 3 rows, okay? Supposed to be 3 rows, and 2 to 2, for example. While I'm doing this, I try to match the so-called inner dimension, so make sure this is 3 rows, that's 3 columns, so the first number should match this, the second. Okay? So this is a good example. It's well designed because it has a 2 by 3, 3 by 6 dimension. A lot of examples online, they use matrix 2 by 2 and to illustrate. That's really not good because when you have a 2 by 2, you can put whatever the same dimension of the row and the column. So this is a good, well-designed example, okay? Here is you and your roommate. So I'm going to immediately translate this into single-cell word language, okay? For example, you are a cell type, which, so let's see, you are a macrophage. I hope you know what macrophage cell is. It's immune cells that will kill others. And your roommate is another immune cells, easy, the B cells. Okay? So I try to make this using examples in the single cell world. Then later you will tell me, so when I link this to that, what that means. So what is 3 here? These 3 are the sandwich type 1, 2, 3, right? So this is the sandwich. One, 2, 3. That's where it's linked. So what's inside here? So that's a butter, tea. So those are the, what are those? Those are the ingredients of the sandwich. So let's copy the 400. What's the number? There are 3, 2, 4. So this is your B matrix. So this is A matrix. This is your B matrix. Your A matrix has a dimension of 2 by 3. And your B matrix has dimension 3 by 6. So this 3 equal to this 3. This is required. If you don't have this match, there's no way you can do this multiplication of 2 matrices. If you type one matrix A and B in the wrong dimension, you put in the MATLAB, you multiply it A by B, it will give you an error message, so you just cannot do it. Okay? So what the 3 means, you have 3 different kinds of sandwich. And here is how many sandwich 1 in this, how many sandwich 2 in this. And in this side will be in each sandwich 1, 2, 3. How many butter? That's a P. What the P stands for? Peanuts, bread, peanuts, butter. So each different components, jelly. Okay, let's translate this bread, peanut butter, jelly, whatever, into the language of the single cell. So what they are? This will be the gene 1, for example, gene 2, gene. I can give a real name of gene, but in this case, we just use gene 1 to 6, 3. Now we need to translate the sandwich into some language which we are going to use in single cell study. So what this should be? This should be so-called gene program. Let's see. Gene expression programs. It could be some gene co-expressed in the same pathway, or they are active together in order to achieve some biological signaling or controlling or something, other functions. So very general language. So you can see, okay, gene program activity. So it's basically some function modules, active modules in the cellular of the two different cells, okay? So the number of this indicate how strong this particular gene expression program 1, 2, 3 will be important or in the macrophage and T cells. So in macrophages, you need 4 fraction of gene expression program 1, 5 fraction of gene expression program 2, and 3 of gene expression program 3, right? And this ratio or different strengths will be different in different cell type. In the B cell situation is 3, 3, 6. Okay, so this cell is more importantly registered for gene expression program 3 in B cell compared to macrophage. Macrophage only needs 3 levels of activity in gene expression program 3. Okay, and then this gradients tell you what's the gene expression level of each different genes that get involved in different programs. So in order to achieve gene expression program 1, we need a gene 1 to be expressed level at 2, gene 2, 4, gene 3, 4. We don't need the rest of genes. Or top 3 genes expression level as a 2, 4, 4 will be important or make this gene expression program 1 work. So you see, when you do the product of this, you start to link the register gene expression program of your different cell type with which gene should express to what level. Okay? So when you compute the first position here, so by the way, so if you have a 2 by 3 dimension A 2 by 3 times 3 by 6, your end product will be a matrix which the inner product will cancel out. We will get 6 by 2 by 6 dimension. So just make sure you know this. And then if you put these two matrices in this position, you will automatically get, because this is 2 rows, and this is 6 columns, so you will automatically get a 2 by 6. This is a 2 by 6, this AB product. Okay, so we need to fill in 12 numbers here, the first number and the second number. And this is easy. You calculate the multiplication of 2 by 4, 5, then you add them together. So this should be 8 plus 10 plus 6. This should be 24, right? And then you do the same thing for this one, but this time you use these 3 numbers to multiply the 2 to 2 and end 6, 6, also 24. Okay, so what I'm doing here is to get the GN1's expression and their involvement in the 3 programs, and weighted by how much each program I need this gene. And then when I put them together, I get the number here. So this number 24 indicate in macrophages, I need gene 1 to be expressed level at 24 if I look at it globally, without knowing what 3 gene expression program looked like. In my macrophage, this cell, I get when I measure the gene expression of gene 1, I get 24. I get just a number of 24. I don't know internally, this 24 has been gave 4 fraction into program 1, 5 to program 2. I don't observe that because they're all latent variable. They're all hidden variable, hidden program. I don't know how many gene expression program macrophage cells get involved in. Maybe it's 250. I don't know the numbers yet. That's a biology. But this here, I just teach you how to do this multiplication and what that could be meaning in there. So you do the same thing for the second column here, and then do this, that. You fill in 2 numbers here, and then do that again. So let me just finish this. We take a break. (16, 12, 15, 22, 30, okay? So next week lecture, I will write this, and one of you will come up and fill in those numbers. Maybe take a turn, each one of you will fill 1 column, and we have 6 people. Okay? Just make sure you know how to do this. And I hope you can explain later if this is a situation, not yourself and your roommate doing sandwich, what the macrophage or other cell type are working, get involved in the different gene expression program. Each gene expression program require different combinations of strengths of expression of genes. Okay, describe this whole thing in your language rather than sandwich. Okay, okay, take a break, 5 minutes. Okay, let's get back to this whiteboard here. In real situation, you may not know in your sample, for example, you use facts of fluorescence to get a population of macrophages. So you don't know what they are involved in, so you don't have this matrix. You don't know how many gene expression program every cell get involved. You have this one sample, this another sample. You know, you measured the gene expression in the same, this sample, gene 1 expression level is 24. Gene B, cell B population have also 24 for the gene expression level for the gene 1. So you don't have all this. This is your observation you get from your sample and your average expression of all the genes. Second gene, or you look, okay, this gene expression level is a little bit high in second, in the first macrophage population, but not in the B cell. Well, B cell have a gene number 5 expression level higher than that. You can do the significant test. This is a fold change, okay, double the expression levels. That's all the information you have. But it's more complicated than that because you don't know what gene expression program this cell population get involved and what the contribution of each individual genes to that particular gene expression program, right? Is there any way we can do this so-called opposite operation? So if you know this and that, two matrices, and it's very easy for you to get this, right? But if you think the reversed problem, if you get given this, can you work out as a spy and look, okay, you come here in the shopping mall, you buy 24 bread, your roommate also buy 24 bread, sliced bread, you buy 16-ounce peanut butters, and your roommate buy only 12-ounce. And would you be able to figure out what you and your roommate are trying to do? So that's a problem in the single cell analysis. We try to do so-called deconvolution. I don't know my spelling. It's maybe O deconvolution. Try to deconvolve, to separate this and the product of the multiplication back to their original components and the fraction of each contribution of each components. This can be done in particular way if you have some idea, you have the range of all the numbers should be non-zero, so you have some boundary of where to search. Basically, you need to search all the values in these two matrices. But if I gave you partial information, for example, I gave you this, and from reference, I gave you some of this previous knowledge. I know gene 1 get involved in program 1, 2, 3. Gene 2 get involved in the gene expression program 1, 2, but not 3. So I have the partial information of here. That will help you a lot to solve this deconvolution problem, partially solved. You can also work out this matrix as well using this end product and the partial information here, or other way around, okay? So that will be something we will discuss later of those lecture series, okay? Okay, back to the matrix times vector, okay? So in this case, we had 2 by 3, and this is 3 by 6, and you know it has to be matched, so-called inner dimension. But if this is only 1, this becomes vector. As long as you have this match to your inner dimension, you can do this. Just get rid of this, and you will obtain this vector multiplied by this matrix. You get this. Okay? This is a very special case, but the same operation, same way, just write in this way. You can get. You can also put 6 by 3 here, so you transpose that, and then you have a 3 by 1, then you will get 6 by 1 vector at the end, right? So if you put as long as you match. So if you know how to do this, you should know how to do the rest. Well, let's see what's that mean if you have a matrix A, see, this is a small squared matrix. And then we are doing a vector, matrix to vector multiplication. So my matrix is A, which is a squared matrix 2 by 2. Then I want to multiply this by x. So x is vector. So my first x is 1 by 2. First, we have to know whether we can do this multiplication. So can we multiply A with x? The dimension here is 2 by 2. That dimension is 2 by 1. So you can do it because this is a 2 by 2, and that is 2 by 1. Okay? So-called inner product match. So at the end, we will get a vector 2 by 1. Make sense, right? The same as matrix by matrix multiplication. So what I'm doing will be this to match and then sum. So that will be 5. And do the same thing here. What's the number? 8, 10, right? So that's the product of this multiplied by that. This one here. If I have a vector 1, 1, so I replace this. Then my result will be, so what's that supposed to be? 3, 6. Okay. So the second point, this point, is a more generalized point to showcase what if I use this as a rotation, as an operator. So now I treat this as operator. A matrix as a So now I treat this squared matrix as an operator. So this operator will do something for the data points in the space. So when I apply this as an operator to multiply a data point in the space, I will change the position of this original position of this data. The original position of this data is 1, 1. Now it becomes 3, 6. 3, 6, what that means is this vector become longer. And also the direction changed. So it was 1 somewhere here. So if you can see this clearly, and then after multiplication, it becomes longer, becomes 3, 6, and also rotate in a small angle positively to that direction. So as an operator, this do something on the data points in the space. Do what? Make it longer and rotate the direction. So this is a transformer. This is not the transformer where in ChatGPT is so-called, but that's a operator, okay, to do something for the data points in the space. And the 1 as a general representative data points in the space. You can try another, for example, 3, 5, and this operator will also change the magnitude of your vector and also rotate it. However, this is a special vector. This is a special data point. Because when you using this operator to this special, very special data point, it becomes 510. So when you apply this operator in other data points, all the data points become longer and rotate some direction. But for this particular point, you only make it longer. No change of direction. Okay? So that's where this before and after. So now we have, we should have a good different concept about the multiplication of matrix to vector. So the matrix, especially this squared matrix, can be considered as an operator. It will change the magnitude and also direction of vector, but in the space, there's some special position which, after you apply this operator, this particular position, the representative vector will not change the direction, but only the magnitude. The other way around, so this vector will tell you something about this operator. Okay? We apply this to this special point and not change the direction, but the other way around, this particular point or this direction will also tell you some features in this operator. Okay, this builds up a system called eigenvalue and eigenvector system. They, this eigenvector tell you some feature of this squared matrix as an operator, what it is, the feature of this is. So this is a general concept about the eigenvector. So this is called the eigenvector of this matrix. It's a set, okay? When we talk about eigenvalue, eigenvectors, we are talking about a matrix of squared matrix. It has to be squared. So 2 by 2, 3 by 3, 4 by 4. If you have a 2 by 3 matrix, you cannot do this eigenvalue, eigenvector operation or calculation for its eigenvector. And this vector is called this matrix eigenvector. And the value of this 5 is so-called eigenvalue. And this is eigenvector. So all this are regarding to with respective to a So we have a system of the vector. We have lambda. Basically, people put a lambda here. Okay. So we will gradually later to I review the applications of this system. Now it's just interpretation, the idea about this geometric interpretation of what is eigenvector and what is eigenvalue and what is the matrix, square matrix as operator. And eigenvalue and eigenvector will tell you the feature of this. Okay? I think the immediate question is if I have this eigenvector, eigenvalue system only applicable for squared matrix, whether it's very useful to the general data matrix, because general data matrix, you cannot guarantee you only have squared matrix, right? You have several thousands of cells, several thousand genes, but you cannot guarantee the gene and cell are the same. So why this eigensystem is useful is only start with a squared matrix. The reason is your D matrix, the data, your D matrix. So D matrix can be different dimensions. So this can be n by m. So this is data, okay? Your data. We can easily transform this into distance matrix, n by n. Well, what's inside here is you compare your samples. So the first sample compared to second, and then you do the pairwise comparison, right? Then you get an n by n matrix, a similarity matrix, or distance matrix. So the same sample first compared to itself, the distance should be 0. So you easily get a squared matrix out of your data matrix very quickly, because we care about one data point against another, whether they're similar to each other or different a lot. So we transform this to a distance matrix or similarity matrix. In that case, after this transformation, we get all the information between relative position of those data. And we know the relationship between them. Then this can be used in the eigenvalue system to understand the similarity between data points. We will learn from here by learning its eigenvector and its eigenvalue to know the relationship between data points from this squared matrix. Make sense? Okay, so later on we will use that system to learn the relationship between different cells, or if you consider genes, you can do M by M also, whether this gene is similar to that gene in this population of cells. Make sense? How this system, although it's limited to square matrix, but with some little efforts we can apply to there. Okay. So I think this is just a very limited introduction and very quickly for you to have some quick idea what it is. And gradually we will see applications and in different occasions. Okay, so to do this. So the next will be the inverse of matrix, very quick. And the square matrix you can do if you inverse it, and these two multiplication of these two matrices will become an identity matrix. It's useful, especially you want to deconvolute one of these, so you want to have A times B become C matrix, and you want to do C divided by B and to get A. So this you need the reverse of the B in order to compute C times B reverse in order to get A. So this is just some definition of those. And the trace is to put all the values in the diagonal position. So you just sum all these values. In this case, the trace will be 0. The trace is the sum of all the values in a M matrix diagonal position, the KK. So I 1, 1, 2, 2. That's the definition of a trace. The determinants, I think we can ignore this very quickly. The meaning of the determinant as a transformer formation. You change, basically you change the area and after your transformation of this as an operator, you apply to the vectors, and then after that you change the area under this the space. Determinant for values, the 3D, so just skip that, so we don't need. We come back to eigenvalue and eigenvector official definitions, okay? Eigenvalues and eigenvectors. By the way, this is only one eigenvector. There another set, so sometimes you have two different values, or both will use. Remember the squared matrix A. Okay, A has to be squared matrix. And this system, so they always come together as a pair. So this 5 and this vector 1, 2 is a set for this matrix. This set to determine which of the linear transformation geometrics a particular matrix represented. So this set will tell this as an operator, as a matrix A, will do what transformation? What kind of transformation to other points? Okay, it will make 5 times longer, but then in that direction defined by this vector. You know the vector can give you the direction, especially if you normalize this into the unit vector to make it a lot shorter and let this vector to land it on the unit circle, that will tell you the direction. So this set will tell you the magnitude change and which direction will not change. So this gives you the idea about as operator of matrix A, square matrix A. There it's features in the transformation geometrics. Okay? What does their linear transformation look like? So you always see the different ways to write this. So this just need to get familiar with. So M basically is the matrix A. So in that case, the writing is M times X. Lambda is this value times X. So X is the vector. So translate that formula to our case, we have A times V, okay? Lambda times V. Those are the multiplication between matrix with vector, scalar, vector, okay? So if you do this, you will find that they are the same. So you probably always see different form of this representation, but as long as you know which one is matrix, which one is vector, which one is a value, okay? You will work it out, the same thing, just using different notation. As explained, by most vector will change both direction and length. It's a general case if you apply this transformation or operator M. But those only change in length are called eigenvectors, okay? So we have only one vector, but the system may have another. So if you have two vectors put together, this V become a small matrix. And this can have two corresponding eigenvectors, and this lambda can be a vector contains two values, two different eigenvector values, eigenvalues. There are, I think that's another example with two eigenvalues. So this is our operator, the A matrix square matrix, and this is the first eigenvalue, and this is second eigenvalue. This is formula. So this is the first eigenvector, which corresponding to the first eigenvalue. This is the second eigenvector we can find corresponding to this. So if you do the operation, so in this case we apply this A transformation to the second eigenvector, and you will get, so do this multiplication of matrix by a vector. You do this, you do that, at the end you get this point. This point is just doubled of the original eigenvector, and its eigenvalue is 2. Basically, it makes this eigenvector 2 times longer, but no change of direction. So if you apply to this data point eigenvector 1, you will get -5 as a corresponding eigenvalue. Okay? So this is a squared matrix, and also it's an example of the symmetric matrix. If you transpose this, and you get the same thing. So this is called symmetric because the off-diagonal position are the same. So you transpose it, you get the same. This is called a symmetric matrix. Eigenvalue and eigenvectors for symmetric matrix have some really good features because it guarantees all the values here are real numbers. So you only deal with the complex numbers. Remember, you have some numbers plus small number times I, so those are complex numbers. So if you have A is a symmetric squared matrix, it's a good system. You apply this, especially like this case. If we define the definition of the distance, So your distance from 1 to x the same as x to 1. So this is a symmetric matrix. So it's very good for application of the eigenvalue systems to this squared and also symmetric matrix. How can you compute eigenvalue, eigenvectors manually? So there are some ways you can do it manually if you have a small matrix like a 2 by 2, maybe someone can do 3 by 3. But in general, we use computer algorithms. There's another field of what the field called, the numerical computational field. People in that field will study how can we quickly or accurately compute eigenvalue, eigenvector out of this given squared matrix. Okay, it's not what we can do here. We just use the package called the function, but we know how to interpret it. And here, a small example to show you this. You can do it by hand for small cases. So if you move around this, and it becomes, so this equation, for example, here. If this relationship is hold, you can move around by move this vector to that part of equation, make this becomes V transpose times lambda. Maybe it should be inverse. So basically, you move this to there. There are some operations you can do, and you obtain a transformed version of this. This looks like not operation, not the link, but it becomes a decomposition of A matrix. So you take this apart into some components, and those components are smaller than A itself, but the product about one against another, the multiplication of them become A itself. So basically, you are doing a big observed matrix, and you put the small parts, you decompose that into small parts of vectors, values, combination, multiplication. Okay? So this is one of the applications of this system here. If I gave you a matrix like this, you can decompose them into vectors, values, and then you can sort those vectors based on the values. Then you can reconstruct. For example, you decompose this into those, and then you remove the noise part and reconstruct become A bar, something like this, because you're doing this and throw away some noise from those values and reconstruct. This becomes a denoised version of your original. We have some applications later for this. Okay, so the decompose and the create original data, something like this also. Okay, so we don't have to go through this because we already had that before. But very quickly, I just go over this. So droplet technology, and this is a place you load your samples, partition OU, and by the way, this is not the only system by 10x. Now 10x has a so-called flex kit. Uh, 10x. Seems slightly different from this traditional or original design. So this offer a cheaper solution for some of this more sophisticated cell type. Some cells you just cannot capture it very efficiently. For example, the hepatocytes. If you have a liver tissues, suppose you have a lot of hepatocytes in there, but for some reason, those cells just cannot be captured very well. And if you do the liver tissue, you get a lot of immunocells or endothelium cells from the sample, but very few hepatocytes cell. So 10x Genomics have a new solution called FlexKit and have the solution for those special cases. Some more technical details if you do this manually. Again, I wish you can come up here to explain to people which don't know the system how this barcode, the gel bead covered with barcode uniquely for each unique gel bead, and also what's the UMI, the small system indicate this uniquely transcribed mRNAs. So this is a constructed library, and this in the sequencing facility, the sequencer will read this is R1, that will be R2, okay? R1 contains the 10x barcode to tell, okay, this transcribed from this cell, and then UMI tell you this is not a duplicate copy of that particular mRNA. And this part, the R1 will tell you, okay, this gene is from, this fragment is from which gene, because there are extron fragments inside here. And this because of the pair, so you linked with this, with that. Okay, so the very general principle. Yes, before the original, you have the I, another system, but now we don't need that. This is a gel bead barcode. So if you open the original file, you will see all this. The green part, in the diagram, and then it ends up with here. So all the combinations of the gel bead barcode is using. Is more than enough, because these 16 base pair combinations each are different uniquely, reach to the 737K. So this is the original design by the 2016. I open the file and take a screenshot, and then you can see those barcodes here. And then this part is after the 10x barcode, then you have the unique, this is version 1 UMI barcode. So each of these in the gel bead will labeled with all this next to it. Okay? So you have a controller, you have cell ranger. Now we use the cloud to do the cell ranger, and then we don't need this, because this we get the output from the cell ranger and put in the MATLAB our own package. But if you want to use this, their output you can put in there as well. Pipeline explanation. There are R1, R2, and index files. This command line, if you use Linux system rather than the cloud solution, then you have to know this call this function, which had been installed in your Linux machine, and count, and give you the ID and where the reference genome is, the annotation, and also reference genome FASTQ. That's a genome sequence, either human or mouse, and then how many cores and memory you use to do the mapping. This is the barcode system. Different colors tell you